Uh, let me start sharing. Can you see my screen? Hopefully. Okay. Before I start, textbook. I'm not so sure. I have not studied these from textbooks, and also I'm not sure if there is a textbook available just for omics because this is kind of a fairly evolving field. So I'd suggest internet resources are the best to study these kind of things. Uh, so the companies that use these technologies have kind of pages. So, for example, Illumina will have a lot, kind of a they have their own websites where they kind of describe all these processes and the chemistry in detail. So I would suggest reading it from internet sources only. Textbook I am currently not aware of. I'll talk with sir and see if we can suggest something. That is the case. I'll in the next class I'll give you the suggestion for the textbook. Okay. Now before we start today's module, this was the question in the last module that was an issue for all of us. I personally also couldn't solve. So there's one trick to this question where they have asked for the genome assembly size, not actually the size of the genome. It's the genome assembly size where you do not need to take care of the gaps. The genome assembly size is basically the sum of all the contact lengths in the genome. So basically, you can you can think of this as a redundant information, and like you do not need to consider this while calculating the genome assembly size. Just the contact lengths and the total of the contact lengths will give you the genome assembly size, which I think is the first option, if I'm not wrong. So you can try it yourself. I think it will come as the first option. So the trick being that they have asked for the genome assembly size, not the genome size. So it's just the addition of all the contact lengths in the in this case. So I hope this answers last week's doubt. So yeah, I want to start off with this. Okay, so coming to this week's module, so uh, we'll be talking about different sequencing methods again, continuing from the last one. So last week we talked about Sanger sequencing and sequencing by synthesis. I talked a little bit about Illumina sequencing. We'll continue the Illumina sequencing part in future classes because there are modules specifically there for Illumina sequencing. So today also we'll be talking about different sequencing technologies after Sanger sequencing. So yeah, so let's get started. So the first one is basically pyrosequencing. So Roche 454 is basically the brand name for the sequencing method, but the technology is basically what is known as pyrosequencing. I touched upon it a little bit last class, but I think in this week's module it has been discussed in much more detail. Okay. Okay. Yeah, initially this was developed by a company called 454 Life Sciences, which was later acquired by this company called Roche Diagnosis, and they kept on using this technology for a long period of time. So the idea is that you'll have DNA inside small water droplets or in oil droplets, which is basically emulsion PCR, and with each droplet containing a single DNA template attached to a primer coated bead, which then forms what is known as a clonal column. And the sequencing machine contains many such wells for parallelization, and it uses a specific enzyme called luciferase, which is used to generate light for individual nucleotides added to the nascent DNA. So the concept is similar to one thing to notice before I move on is basically the sequencing technologies that we use, the various sequencing technologies that we are using and formerly have used. So this is again like pyrosequencing is something not widely used today because we have much simpler, much better technologies with Illumina and long read with Oxford Nanopore and PacBio and things like that. So these are kind of legacy technologies. We do not use them in real life right now. But in some cases, where if the sequencing costs are low, we use this legacy technologies. But generally, these are not used. If you see and read recent papers, you'll not find these technologies being used very frequently. But then again, we need to study these because we need to know the evolution of these technologies to understand from where we started to where we are now. Because where we are now, we are at a very good position to generate a lot of data with consortium projects like big consortium projects like 1,000 Genome Project or the UK Biobank or uh, similar projects like Genome Asia or etc. So where we use these kind of technologies, not phyto sequencing, but Illumina and above, to kind of sequence thousands of individuals together at a decent cost, which is not possible with the legacy technologies. But the chemistry is kind of similar by the idea of either you do sequencing by synthesis or you do reversible termination of these. The basic idea is sequencing by synthesis, and on top, either you do irreversible or reversible termination, depending on the technology. But yeah, so the idea is that you'll add a nucleotide or a base, and it'll emit some signal in the form of light or in some cases in form of some radioactivity, which can be captured by a secondary device, which can translate the signal to the fact what nucleotide has been added. So in this case, in pyro sequencing, we use something called the luciferase to generate the signal, which is actually a fluorescent light, which is captured by some camera for later use for signal detection. Actually, what base has been added uh, to be mapped to the signal that has been emitted uh, by this particular enzyme. So the read length of these technologies is around 200, 300 bases, not far away from Illumina. Illumina is around similar times, slightly more in some cases. But in today's world of long read technologies, this has gone up a lot. This is for now, I think ONT, Oxford Nanopore Technologies can sequence up to 10 KB and even more. But again, the longer you sequence, the higher chance that there are error rates. The error rate goes up because obviously for every base sequence there is some probability of error associated with it. But again, yeah, so 200, 300 bases is not a very bad Greenland, but still it is lower than what we expect kind of today. So it is around like 100 MB of output run, and a single genome can take about 7.5 hours to run. Now, what is the principle behind it? So, the first step is basically nucleotide incorporation. I'm just talking about the sequencing part, not the DNA preparation and the pre-processing part. I'm just talking about sequencing part. So what happens is the nucleotide, first step is the nucleotide incorporation, which is basically a DNTP present in the solution. Will be added to the 3' end of the DNA strand, and which releases what is known as a pyrophosphate as a byproduct. Now, ATP sulfurase is an enzyme that will be present in the solution. What it will do is it will catalyze a reaction between this pyrophosphate and adenosine 5 phosphosulfate APS to produce what is famously known as ATP, which we know as the energy currency. And what happens is during this process, the luciferase enzyme that is present uh, will use this generated ATP and luciferin, which is also present in the solution as substrate, and and will convert luciferin to oxyluciferin, which will release a visible light, which will be captured by a camera or some secondary device. And the intensity of the light will tell you, which is basically directly proportional to the number of nucleotides incorporated. And after the detection, there's another enzyme called apidase, which will be introduced to degrade any unincorporated DNTP so that in the next cycle it is not contaminated. So the idea is that you add a nucleotide, it releases pyrophosphatase, and this pyrophosphatase will bind with another compound to form ATP, and luciferase in the system will use this ATP and luciferin, another substrate, to combine and give something called oxyluciferin, and it will release the light which will be captured. So, uh, uh, s- okay, this is the general principle of how the pyrosequencing works. If we go into details, this is something that is more focused on the pre-processing part of the sequencing, basically how you do the amplification and strand separation, and you set up the sequencing experiment. So what happens is in case of amplification, you do PCR with biotin labeled primers. So one primer will be biotin resulting in PCR products where one strand will be tagged with biotin, and this biotin um, will be captured with streptavidin beads, and this streptavidin can be used kind of interchangeably with other compounds as well. There are other compounds also which acts similarly. It is basically just holding the bead in one place, and then the biotinylated PCR products are immobilized on these streptavidin coded magnetic beads, and, which anchors one the DNA fragment. Yeah. Now, then next step is basically denaturation. So you have to like kind of convert the double-stranded DNA into single-stranded, where this alkaline denaturation is used with NaOH as the substrate. And if you have enough alkaline, if the pH is high enough, what it'll do, it is break the double-stranded DNA into no, single-stranded DNA, and the non-biotin complementary strand will be released and removed, while the biotin labeled strand will be present with the bead holding on to it. Then again, the next step will be the washing step, where basically you remove all the unbound primers and the excess reagents, and you only are left with the single-stranded DNA template, which will be there for sequencing. And then during sequencing again, sorry. I think. Just a second. Let's stop presenting for a second. I think something may show code. Sorry. Yes. Yeah, during the sequencing step, as we discussed, you have this APS substrate along with luciferin, and the pyrophosphate that is going to be released will bind with the APS, and it will convert to ATP, and luciferin will use this ATP to give you that signal. And you will have to provide with these enzymes inside your solution, which will be DNA polymerase, ATP sulfurase, luciferase, and apidase for the last step to kind of remove all these extra all these signals for the next cycle. right? So this is kind of the pre-processing step. You have the streptavidin coated beads with the DNA primers, and then you tag the biotin in one of the strands, then you use NaOH to kind of break up the strands, and basically this strand will be washed away. And if you take this strand and then kind of use the sequencing by synthesis method, where you have the DNA polymerase, a short primer, and the DNA polymerase to keep on synthesizing this strand and basically sequencing the single strand. Okay. Uh, So yeah, after uh, this process is like during this process is occurring, uh, inside the machine, the primer will be first hybridized on the template, and then gradual DNA synthesis will occur. Then the primer will keep on adding one nucleotide after another. Uh, and therefore the reaction will take place with release of pyrophosphatase. And if the DNTP added is complementary to the next phase of the template, it is inserted by the DNA polymerase, and this pyrophosphate molecule will be released. The light production will be recorded during the entire procedure by means of a camera. Then it is quantified, evaluated, and presented in form of signal peaks. Right. So there will get a graph basically with peaks showing the intensity of the molecules. Sorry, the intensity of the light, and based on that, you can detect what base is at that particular position. Uh, If two or three of the same nucleotides follow one another in the template, several nucleotides will be installed in a single round, and the light production will be two or three folds high as normal. So basically, if you have if you have a repeat region in the genome where you have like five A's together, the proportion of the peak at that position at that five positions together will be kind of proportional to the number of repeats there. So if you have five, you'll have a certain peak. If you have 10, you'll have like twice that peak. So it kind of is proportional to the intensity is proportional to the number of same nucleotides, one after another. So, and if no nucleotide is inserted, it will remain black and no light signal will be emitted. And again, in the last step, to remove surplus DNTPs and complete the reaction rapidly, the DNTPs can be broken down into DNDP, which is basically diphosphate, and break, you turn the DNTP into DNDP and phosphate by using this enzyme called APIase. So this is a schematic of the same. You have this uh, this is the primer, then this is the DNA polymerase, which is kind of adding the nucleotides one after another. And after one is getting added, this phosphate molecule is getting released, which combines with the AMP to form ATP. Now, this ATP will use luciferin, and the luciferase enzyme will convert this to a light signal, basically oxyluciferin, and this basically light signal, which is captured by these particular peak signals, which you can deduce what kind of pieces are at that position. Okay, so yeah, this is how generally pyro sequencing works. This is how generally pyro sequencing works. So yeah, I hope I have given you an understanding of how this works. Uh, Nowadays, again, as I said, not very widely used, but still a very good technology, and this is one of the next generation technologies. Uh, Along with Illumina is kind of further down the line, but this was one of the earlier next generation technologies. Now we are at third generation, right? Okay. So how the timer extension occurs is basically, so again, this extension is, as you know, is basically one after another, and a particular base getting released. Yeah, this is NGS basically. This is what is in NGS is, but right now we have kind of surpassed NGS. We are in third generation. So again, Illumina is also NGS. This is also NGS, but We have kind of moved past those technologies. Not Illumina, but these, at least the pyrosequencing part, we do not actively do now. But yeah, this is NGS only. This is not like the older generation technology. Uh, Now, yeah, the addition of these nucleotides can be in two manners. One is basically a reversible this termination can be reversible. So again, when one base is added, one signal is emitted. Either that can be like one base added and then the reaction stops because further it cannot go, or it'll be like one base added and the modification is removed, and then you can keep on adding the bases. So that is what reversible termination is. So the idea is that the polymerase that came into the picture started the catalytic reaction using the 3' hydroxyl group at the end of the initial primer. It incorporates the first nucleotide, uh, then it extends the primer at one nucleotide addition per, per go, right? The nucleotide then contains a pseudochrome and a blocker or a terminator molecule for detection and termination respectively. A terminator is present probably at the 3' end and blocks further extension. If you remember the Sanger sequencing technology, that is how it is done. One base added, if the perfect base is added there, then further downstream sequencing won't occur because that's a DDTP. Sorry, yeah, dideoxy nucleotide triphosphatase, and generally what we have is deoxynucleotide triphosphatase. So the extra 3' O OH, the uh, OH will be converted to a O, which is the deoxy method. So that is by dideoxy. So uh, once the reaction is blocked, it is blocked. You cannot kind of continue further sequencing using the same strand. You have to use a separate strand to continue the synthesis. But there can be cases where you can do this combination, this addition of the nucleotides, then release of a signal, then again addition of the second nucleotide, like that. So that is what is known as that is what is known as reversible termination, right? So before detection, the first round of washing is conducted, and this will remove all the unincorporated unused nucleotide to avoid signal disruption. Now, the fluorophore is released from the nucleotide, and using a laser light, this fluorophore is emitted and it is captured using high definition camera. Now, once this has happened, either the reaction will stop or you can somehow remove the fluorophore and then you continue with the reaction. So you can either stop the reaction or you can remove this terminal signal and then keep on adding nucleotides one after another. You add one nucleotide, you get the signal, then you again reverse this termination, and then you again add another nucleotide, right? So immediately after the fluorescent detection, the terminator or the blocked molecule will be removed by the washing of the nucleotide, and the 3' hydroxyl is ready for another round of synthesis. Because in earlier Sanger technologies, what we used is a DDTP where you do not have this 3' hydroxyl group present. It will be deoxidized to a it will be deoxidized to a double bonded oxygen. The hydrogen will be removed. So, if you can somehow free up this 3' hydroxyl, this will allow for further primer extension. And the process will occur in the cycle for all four nucleotide incorporations. And lastly, all components are then washed off from the flow cell before another round of nucleotide incorporation, right? So it is basically massively parallelizing all the four nucleotides together. So here you are detecting only one kind of nucleotide because the signal is very specific to this nucleotide. And once the T is added, you cannot further add anything more. And once the T is added, you know that at this position, T is added because of the signal that got emitted. Now, what you want to do is basically continue doing the sequencing one after another, one base after another, in a reversible termination manner so that you can get the sequence at one go. Uh, Yeah, so the modified nucleotide analog, which will contain the fluorophore and also the reversible terminator, when added in a complementary location of the template DNA, will temporarily block the primer extension until the fluorescent signal for the nucleotide incorporation is detected by sequencing by synthesis chemistry. And after the detection, the fluorophore and the blocker will be removed and washed off from the flow cell to allow for a second round of nucleotide incorporation. So uh, this is how it looks like. So you have, this is the template DNA. This is your DNA polymerase. This is the initial primer. Then you're adding one base after another. So one base has been added. Fluorescence has been emitted. You have captured this using the camera, which is a detector. And then you have to use a reversible terminator, which can remove the blocking. Right, whatever be it the dideoxy part or some other kind of blocking that is blocking the another nucleotide from getting added. You remove that blocker, and then you kind of add another base, get the signal, remove the blocker again, add another base. So this is what is known as reversible terminator. So before moving on here, so uh, again, we will be talking about further different type of technologies, but I want to tell you just one thing, keep one thing in mind. We are all talking about sequencing by synthesis. The general idea is that you have a double-stranded DNA, you make it single-stranded. This is actually applicable across all SBS technologies, right? You take a double-stranded DNA, you break it down into single strand, you have a primer, you have a DNA polymerase. Now you have different type of nucleotides in your system, in your experimental setup, in your solution. That will depend how the the bases are there, what kind of bases are there, and how modified those bases will be depending upon your choice of the technology. But the basic idea is that you have to detect after one base is getting added, what base is that. So for that, you need either fluorescent signals, which is the light signal, or you will use something of a radioactive signal or something else. Okay, so this is the basics of the SBS chemistry. I like whatever sequencing technology you study, you remember this is the basic. This is how it is generally done. So you can build upon this. You can introduce much more complexities into this, how you're detecting the signal, what kind of signal you're detecting, those kind of things, reversible termination, and this kind of things. But these are fancy stuff, modeled on the base chemistry, which is sequencing by synthesis. So understand the sequencing by synthesis well, so you'll understand the further technologies better. Okay, the next type of technology we'll be talking about is ion tolerant sequencing, which is also the pH sequencing that has been discussed in the course module. So, in this case, again, you see the sequencing by synthesis chemistry is intact. It's just the mechanism of detection has been changed. So, what happens? You have a single strand, you have DNA polymerase. When a complementary nucleotide will be incorporated in that strand, a phosphodiester bond is formed and a hydrogen ion will be released, H plus ion will be released, which will change the pH of the medium. In this case, you are not detecting the addition of the nucleotide by some light or radioactivity. In this case, you are detecting by the change of the pH of the medium. Right? The ion sensor will actually detect this in this case, instead of a camera or some secondary device. This will just be an ion sensor which will detect the amount of pH change in the system after addition of one nucleotide, and it will convert that signal back to what base this get. Sorry, I think this is. Am I audible now? I hope I'm audible now. Okay. Yeah, some internet issues probably. Sorry for that. Yeah. Now the idea here is, the idea here is that you are detecting the change. Sorry, you're detecting the addition of the molecule as a change of pH instead of a light signal or some radioactive signal, which is captured by this ion sensor, which will convert the chemical information into digital electronic information. As in other kind of NGS, the input DNA or RNA is fragmented. This time this is around 200 base pairs, so this is the read length. Around we are stuck around that read length, if you notice, 200 to 300, right? The adapters are added, and one molecule is placed onto a bead, and the molecules can be amplified on the bead using emulsion PCR, again similar type to what we see in the pyrosequencing technology. And then each bead is placed within a single well of a slide, and you'll have these thousands of wells in a slide, so that you can massively parallelize this sequencing. Okay, so yeah, this is how uh, it kind of this is how the overview of it looks like. So again, like 454, the slide is flooded with single species of DNTPs along with buffers and polymerases, one NTP at a time. The pH is detected in each of the wells, as each hydrogen ion released will decrease the pH. The change in pH allows us to determine if that base and how many thereof were added to the sequencing read. So one thing to keep in mind is, generally in a single well, uh, these kind of slightly older technologies, what we'll have is they'll have a single type of DNTP, right? Either you have a or a T or a G, whatever. So basically, the change in whatever in the signal intensity, in this case, it's the pH, will tell you in that well if a particular DNTP has been added or not. Okay, so yeah, in this case, the similar type of thing, the DNA is broken down into single strands. Then you are adding a primer and a DNA polymerase, and then you are adding all the DNTPs inside the solution. And after the DNA polymerase adds one base at a time, the pH will change of the solution, and the ion sensor will detect it. Right. Then after that, the DNTP will be washed away, and the process is repeated, cycling through the different DNTP species. So one at a time. So if you take in a single well, if you want to detect what are the A's present, you do A first, then T, then G, and C, like that. Right. So one base at a time incorporation it can handle. So this is how the signal graph will look like. So see a higher intensity of some nucleotides compared to the other. What it means is basically, at that position, multiple of these nucleotides have been added instead of one. Again, the signal intensity is proportional to the number of nucleotides added, as in case of pyrosequencing as well. The pH change, if any, is used to determine how many bases, if any, were added in each cycle. So again, the intensity will be proportional. So next, we come to what is known as sequencing by ligation. Uh, uh, the name of the technology is SOLID, SOLID, sequencing technology. So again, the principle is slightly different from synthesis. In this case, ligation will occur, not synthesis. In this case, instead of DNA polymerase, what we'll use is DNA ligase. I don't know if you remember or not. In the DNA replication, DNA ligase takes a very important role to join the Okazaki fragments of the 3' to 5' strand, right? So here we use the DNA ligase instead of DNA polymerase. So here also we use a signal, which is the fluorescence. We use fluorescently labeled probes to detect nucleotide pairs, not individual bases. Slightly different here, but again, uh, the idea is you have an enzyme, you have a single strand, you have a lot of bases, and you're trying to detect which base is getting added. Right. So firstly you fragment the DNA and you amplify the DNA, which is kind of a basic step across all the technologies. Then these DNA fragments will be attached to beads, and these will be amplified using emulsion PCR. Then probe hybridization and the ligation will occur, and the probes will contain two known bases and a fluorescent label. The DNA ligase will join the matching probe to the template, and the fluorescent signal will be detected. And this cycle will be repeated multiple times to get the whole sequence. At the last, the fluorofluor will be clipped off, and the new probe will be added. And each cycle will start at a slightly offset position to ensure multiple reads per base, which is the you want to increase the coverage. So this is kind of so, yeah, this is kind of the whole process in a workflow. So this is your bead, this is your adapter sequence, and this is your basically the sequence you want to know the sequence of, right? So first, prime and ligate. So this is the primer, this is your template sequence, and you basically ligate these using ligase. This is the sequence primer, and this is the bases that are getting ligated here. Then after ligation, it will release a fluorescence signal, which you can image using some high definition camera or things like that. And then basically you extend cap the unextended strands so that you can block the signal there, and then you cleave off the fluorophore at that position, and then again you repeat steps 1 to 4 to extend the sequence. So you add a couple of bases, the fluorophore emits the signal, you capture that signal using some high definition camera, and then again you wash off the fluorophore, then again you start doing the ligation, right? So how it'll do one cycle after another to get the whole uh, uh, whole sequence. And then after that is done, what you can do is you can reset the primer. You can basically melt off the extended sequence and just get the primer back again. And then again, you can do so. Basically, you can recover the primer back from this experiment, and you can use it for your next well. So that is also possible here. Here also the idea is similar. If you notice that you are extending the nucleotides in some direction, and then you are getting some signal out of it. In this case, this is a fluorophore. And another important difference here is we do not use the DNA polymerase, we use DNA ligase, because there are two bases together, and you want to just join the bases together. You do not want a DNA polymerase to kind of find the complementary one and then add it. It's just the two bases together. You just put it, and the DNA ligase will add it to the strand. If it is complementary only. If not, it will not add. Right. So that is the kind of the difference, and this is how the signal uh, information will look like. This will be the read positions, and basically it will indicate if a in a particular position a particular nucleotide has a group of nucleotides have been added or not, and these are basically different cycles given by different colors, right? So uh, more the cycles, basically higher the number of cycles, the more of the information of the sequence together you can get. If you're going off to like say 20 cycles, in this case 35 cycles, you'll get around 70 bases or some slightly more than that, uh, for each cycle around 2 to 3 bases. So this is how solid sequencing technology works. Again, it is not sequencing by synthesis. You're not synthesizing a DNA strand, you're just adding DNA strands. But again, the idea is quite similar, right? So in this case, we use ligase, not polymerase. Okay, next we come to something known as polyny sequencing. So basically, what is a polyny? First, it's basically a cluster of DNA molecules which has been synthesized using DNA polymerase, and it is kind of a analogy to colony of bacteria in a culture, right? It's similar sequences that have been sequenced. So you have a lot of similar strands together in a place, and you put the DNA polymerase there, and it will sequence those strands. So it's similarly sequenced strands clustered together in a small area, which is known as polymy. Right. The polyny sequencing works on the principle of amplifying and analyzing DNA within a solid matrix or on beads to allow accurate and simultaneous sequencing of millions of fragments. So, uh, sorry. So this method will be using to create tiny clusters of identical DNA molecules, which are known as polymies, which are immobilized in a matrix and serve as templates for sequencing. Each polymer represents a single DNA template, ensuring the amplified sequences remain distinct. This prevents mixing or contamination between DNA fragments, and then sequencing will be performed on these polymers using fluorescently labeled nucleotides. Again, similar concept, fluorescently labeled nucleotides to identify each base in the DNA. The process begins with constructing a paired tag library from genomic DNA, followed by clonal amplification, again on microbeads, using emulsion PCR. This is kind of a step that is followed across different technologies. This amplification produces discrete polymerase colonies that serve as bases for sequencing. Again, like this is very analogous to bacterial colonies. So small areas inside your matrix clustered DNA, similar sequence of DNA will be present, and you can use synthesis, sequencing by synthesis to kind of know what is the sequence at that for that particular strands. So yeah, the amplification produces discrete polymerase colonies that serve as a basis for sequencing. The amplified beads are then enriched, and the enriched beads are then embedded in a polyacrylamide matrix, creating two-dimensional array that allows simultaneous sequencing of millions of DNA templates. So this is kind of the schematic for polymer sequencing process. So your the DNA is break it down into smaller strands. In this case, in some cases, what you'll do is circularize the DNA so that it kind of does not get attached to other strands. Then you use amplification on emulsion PCR beads, and the beads will be immobilized in a monolayer of acrylamide matrix. Sorry for that. And then across different parts of this matrix, you can use sequencing by synthesis to know what is the basis. So what you're doing is you are also, in this case, also massively parallelizing that procedure of sequence of knowing the sequence of multiple strands together. Right. Uh, Okay, so yeah, that is all from my side in terms of explaining the course modules. I think I have left out a few details because I do not want to kind of exactly talk about what Sir has talked about, but I want to talk about a little bit more in terms of the sequencing stuff, not from a sequencing perspective because I am not involved in for my work, I do not do sequencing that much. What I do is the computational part of after that. So basically, after sequencing, what happens in terms of data analysis and generation and analysis. So I'll give you a little bit overview about what happens after that. So just 5 minutes. I have a different PPT for this. Okay, so after the data has been generated, you have lots of these uh, uh, so again, this is not part of your course. I just wanted to give you a certain overview so that you know after the data is generated how it is processed, right? So you generate FASTQ files or you'll generate the read information of different DNA strands. Now you need to know where these strands actually came from in the original kind of um, or original DNA. So you have the whole DNA. How have you sequenced? You have taken the whole DNA, you have broken down into smaller strands, you have amplified it, and so there will be overlap between these reads, right? So after you have sequenced these reads, you have to kind of map the read back to the original genome. So how you do that? That is what is known as the alignment problem. So these are the kind of things that I work on. So the idea is simple, that you have two sequences, protein or DNA, whatever it can be. So you have two sequences and you need to align one sequence with the other. So assume this is one sequence and this is the other sequence. How do you align that? So there can be gaps, there can be mutations. If one base is changed, that those are known what are known as SNPs, single nucleotide polymorphisms, and those basically are mutations, right? So you have to consider those while doing the alignment. So this is what the alignment problem is. So you have two sequences, and what you want to find is basically the optimal alignment between two sequences in the form of a list of columns of different bases, right? And so there can be variations in the sequence: substitution, deletion, insertion, inversion. So these are basically your mutations that can occur in the genome, and this is what you want to kind of check, right? So we have different scoring schemes for that. We use kind of, if A matches with A, we use a scheme. If A does not match with A, it is matching with C, we use a penalty. So we use a scoring scheme to tell if the alignment is optimum or not, right? I'm quickly going through this because these are not part of your course. I just wanted to give you an idea about what happens. So there are various algorithms that can be used computationally. Each one is different, but the basic one is basically brute force. You take, this is your whole string. You have a small substring, which is your read. You want to map it to the whole DNA. You check one after another where it is mapping. So that is what is the brute force algorithm. It takes exponential time, so it is not practical. So this is what is known as BLAST. So I hope you have heard of it. It's a software where basically you can query a sequence into a database of sequence to find regions of similarity, and it also uses a heuristic approach, which is the brute force approach, to do this, uh, making it faster and more efficient. So again, I'll not go into the details, but what they do is basically break up the read into small sections, and what they create is a lookup table with indices. So basically, you know, this small string has come from that, this small string has come from that, this small string has come from that, something like that, in that manner. uh, okay, I'll move on. So this is what indexing and creating substring looks like. Is basically you divide the whole DNA segment into smaller strings, and then you index, and you know where particularly this string has come from. Okay, I'll skip all these. These are a little bit more advanced in terms of computation. So this is the algorithm that we currently use. Actually, this is not state of the art, but this is an algorithm that you should also look into. This is what is known as Burrows-Wheeler transform. So basically, you transform a whole sequence into a smaller set of subsequences, and you want to know where the subsequences actually came from. So this algorithm does that. I'll not go into this. So basically, what you do is you break up a sequence and into smaller sequences, and you connect them by nodes. So this is computationally, this is what it looks like. So you have a root node, then you have other, so the edges will tell you basically what the sequence is and what overlap is there with other bases. So for example, BA, and this is the termination signal, the dollar sign. So this sequence is BA. Again, the top sequence is BA, again ABA. So these two nodes have this particular edge in common. So this is how a tree is formed of the whole sequence, so that if you do this for the whole genome, now that you have reads, if you transform these reads again into the smaller substrings, you can map this smaller substrings from to the tree itself. So what we do is create a tree of the whole sequence, and then when you have these reads of the smaller sequences, we kind of try to map it to them, right? Again, not going into the details. So again, Burrows-Wheeler transform is a very interesting algorithm used for alignment. If you are interested, please look it up. Not going to this. Again, yeah. So what we do is basically, after getting the reads, we want to see if there are variants inside the genome, if there are mutations inside the genome. So as you see, this is the reference genome, the top colored ones, and the gray ones are the reads that have mapped to this region. So you see, it is not a complete match. In some cases, four reads are saying P at this position, two reads are saying C. So this is a possible mutant site. But again, more number of reads supporting for the reference site than for the alternate site, which is the C. So in this case, it is probably not a variant here. So how do we identify these variants? So uh, not go here. Yeah. So for example, these are all the reads, right? These are all the reads that have been generated. This is your reference genome and this is the target genome. So what can happen is at particular positions, again, at this particular position, two reads are saying G, maybe the rest of the reads are saying something else. In some cases, it can be just PCR duplicates. So we need to kind of distinguish between these things is what is a PCR duplicate and what is actually a genomic variant. And yeah, I'll not go into much of these. So again, so those were single nucleotide variants. Now what you can have is structural variants, big chunks of the DNA that have been removed, or big chunks of the DNA that has been inserted or inverted, and things like that. Okay, and actually this is a very important thing if you want to do genomics. The VCF file format is very important. So that is how variant data is stored. So this is how it looks like. It will give you chromosome, the position, if it is a known ID, if it is a known step, what is the reference allele, what is the alternate allele, what is the quality, and basically other information. So this is basically AF stands for allele frequency. AFR allele frequency is basically these are taken from the Thousand Genomes project, where they have like different uh, population sequence: African American, South Asian, and East Asian, and I'm forgetting a few, but like what are the allele frequencies in those populations so that you can compare. And this is basically the genotype, if it is present in one strand or it is present in both strands. Okay, I think that is enough for the extra information. But yeah, I just wanted to give you an overview about how things work after the sequencing has been done. So after the sequencing has been done, we take the data and we pass it through a lot of pipelines. We run softwares through it, we align the data, we find variants, and we try to figure out what these variants are actually causing. Is it causing a disease, or is it not causing a disease? 99% of the cases, the variants will be benign. It will not cause a disease. But in a few, that 1% of the cases, it'll maybe change the structure of the protein. It'll maybe change the sequence of the protein. If you know the basically sickle cell anemia disease, it is due to mutation of one single base uh, inside the genome, and it just changes the amino acid at that position, and it causes sickle cell anemia, where your hemoglobin molecule kind of looks like a sickle cell instead of a normal hemoglobin molecule, and it cannot hold on to the oxygen as a normal hemoglobin molecule. So sometimes these variants can be very effective in terms of changing the protein structure and the sequence, but in maximum cases, this will be benign. So we need algorithms to kind of differentiate between these. We work on those as well. Okay, so again, thank you for listening. Again, now we'll do the assignment problems. Again, you can write the answers in the chat box. We can discuss. Okay. Do we have any answers? Okay. The answer will be that deuterium DNTPs will have lower probability due to two things. Firstly, is due to the bond formation between deuterium and difference between hydrogen and deuterium will be slightly different. The affinity for hydrogen will be much higher compared to deuterium, which will be the heavier isotope for hydrogen. So if you have equimolar DNTPs of hydrogen and deuterium, it's most probably hydrogen will be incorporated and deuterium will be kind of have a low probability of incorporation. It's because of the size difference and as in size and molecular weight difference between hydrogen and deuterium because it's a heavier component. Okay, moving on. Notice the repeats for this question. Yeah, yeah, the answer is 8. So these are the signals that you pick up. The repeats will be picked up together. The intensity will be higher, but it will be one signal, right? So the answer is 8. Correct. Okay. This is a multi-option correct, so there can be other correct options as well. Yeah, the answer is both charged coupled device camera and photodiodes. Basically, you would need a device which can convert this fluorescent signal to some electronic signal or some photo, basically. So the charged couple device camera can do that. Photodiodes are basically diodes that can convert the photon signals, basically the light signal, into some electronic signal. So uh, both of them can be used. The answer is DNA polymerase and DNA ligase. Gap fill and barcode padlock uh, is n- that can be used for linearization, but it is not for quantifying the spatial organization in situ. So for quantifying the spatial organization, you need some enzyme that actually is this kind of synthesizing or adding nucleotides to the DNA. Right? For that, DNA polymerase and DNA ligase will work. This gap fill and barcode padlock probe is just a probe, right? It's not something that is extending or synthesizing your DNA. So technically, that is not the answer. The rest two are. So B and C are the answers. This question, I also have a doubt. So I would like to hear your reasoning as well for this. Okay. The answers are basically the amino group, the allyl group, and the acidomethyl group. So I was not very sure about why the other group is not uh, there, because the only difference between this and this is basically the double bond at the second position for the third option. So what I am assuming is this will cause some sort of hindrance or some sort of steric hindrance kind of a thing that will not allow it to be removed easily. Amino group is basically a well-known reversible terminator. It can be removed through just if you oxidize that position, it will remove uh, the amino group. This is also the 3' OLI group is also reversible. It can be removed by dealylation, which is also a well-known chemical reaction. The acetomethyl group again also it's basically used in Illumina for reversible terminator, and it can be removed by phosphines. But I'm not sure about the third option. I'll be honest because chemistry wise I'm not sure how difficult will this be to remove compared to the second one. I will get back to you on this question after I discuss with sir and his other TA. So probably I cannot give you an explanation about why the third option will not happen right now. But yeah, the rest of the three are well known. So yeah, Swati, I also think so, but I'm not. See, I do not have that chemistry background to actually comment I- in a certain manner. But I also think there's some steric hindrance going on because of that second bond, double bond at that second position. But I'll have to confirm this and get back to you. Yeah, so this one, A and B. Okay, yeah, the answer is correct. So it'll do high throughput because at one go you'll be able to sequence a longer, not a longer read, but all the read together. Read length will not be increased because it is shorter than pyro sequencing. And yeah, the homopolymer errors uh, will be removed. So yeah, the answers are correct. Excuse me. Yeah, uh, the answer is static hindrance. Basically, again, Solexa is basically Illumina sequencing. It's the same similar thing. Uh, they so the reversible terminator nucleotides are modified to allow incorporation of only one base at a time. In some versions of this chemistry, the 3' hydroxyl group is not chemically blocked, but a bulky fluorescent label is attached to the base of the sugar, which creates this steric hindrance. And the steric hindrance prevents the DNA polymerase from adding the next nucleotide, and which effectively terminates elongation even through the 3' uh, hydroxyl group, which is technically available. And once this base is read via fluorescence, the blocking group is cleaved, and basically the dye, which causes the steric hindrance, it is removed, and next cycle of incorporation begins. So the answer is steric hindrance. The answer is all three. Solexa and Illumina are kind of the same thing. So it's the same thing. And Helicose also uses this reversible termination. So this is basically single molecule sequencing. We have not talked about helical sequencing, but again, it's a similar sequencing by synthesis approach, and it uses this reversible chain transcript termination for DNA sequencing. So, and Illumina and Solexo are Solexa are kind of the same thing. Just a second. Hello, I'm a class snitch. Yeah, the answer is basically all three except the second one. Second one is not actually a actual compound, so it's just a false name. The rest three are correct. So yeah, yeah, that's the correct answer. No problem. Yeah, okay. No, have to be a little bit more careful. So 16,500 Daltons, but it's a double-stranded DNA. So for a single-stranded DNA, because individual constituent in the DNA library, when they bind on the magnetic beads, will be single-stranded. So you have to find the first weight of the single-stranded DNA, so which will be half of this, which I think is uh, around, if I'm not wrong, just taking 6, 5, 6, 4. Yeah, 8,250. right? Then you'll have adapters. So there will be two adapters, each of 244 Dalton, right? 244. So you add 488 on that. So that comes to 8705. No, sorry, that comes to 8738, right? Two adapters. But again, there will be another primer attached to it, right? So another 244 will be around similar to the adapter, which you add to 8738. So it gets to 8932. That is the answer. So you have to be careful about this because this is a double-stranded DNA. This 16,500 will be the component of the double-stranded DNA. You have to break it down to single-stranded, which will be 8250. Then you add two primers, 488, and then another 244 for the, two adapters, 488, and another 244 for the primer, so which is 8932. So be careful while doing problems like this. You have to add the primer. I also, when I was doing this, I missed the primer. You have to add the primer, which will be roughly the similar type of weight to the adapter as well. So it's 8932, not the fourth option, but the third one. The answer is both, yeah, both pyrosequencing and ion-ion sequencing, because in pyrosequencing, you do not need fluorescent nucleotide labeling. What you need is an enzyme, which is luciferase, and basically the substrate, which is luciferin. You do not need to tag a fluorescent uh, element or a fluorescent compound to the nucleotide. You do not need to do that. You'll have luciferin inside the system, and you have luciferase also, which can do this, which can give out this fluorescent signal. You do not need to tag it with the nucleotide. So answer will be both pyro sequencing and ion sequencing. Ion sequencing, you'll only have changes in pH, so that is intuitive. But the other one will be pyro sequencing will also come. So for these questions, when you do this, just be a little bit more careful, because some of them are a little tricky. See, go through the options and try to understand, and then you do it. Right? Moving on this quickly, because yeah, this is a no, uh, this is a thing that all the compounds that will be released are luciferyl adenylate, and that will be one of them, oxyluciferin, and it will be all three. The acetylate will not be there, but rest of them, all three will be present after the reaction. So yeah, keep this in mind. Like what kind of reaction goes on, and what are the specifically for pyrosequencing, a lot of compounds are involved, so you would want to remember what the reactions are going on there. No, actually, it identifies in 2, 7, and 6, all three, except for 5. Go for 2, then 4, then 6, then 7. In series 1 ligation. Yeah. No, it will cause a hairpin. So it you can say a hairpin is an internal loop, but yeah, basically, if you have palindromes, in the middle portion there will be a loop which will not be kind of connected, and the end portions will be connected. So it will be a hairpin. It, technically, you can say it is a internal loop, but the idea is same as like RNA folding. You see how tRNA folds with the hairpin loop because of palindromic sequences. It's similar concept here, but yeah, the options are very close to each other. Internal loop or hairpin. It's basically a hairpin if you are being specific. So it'll cause interbase stand pairing and will create that hairpin loop. Yeah, that is correct. So this is coming from that bacillus bacteria. This is quite well known. Let me know. It's the correct answer. And that's the final question. We have not covered complete genomics sequencing, but sir has covered. The answer is around 150 nanometer. So yeah, you'll have to probably remember this. So okay, so that is all from my side. This was the last day question. Again, I'll repeat if somebody was not there at the beginning. So in this case, you do not consider the gaps because the genome assembly is basically all the summation of all the contiguous. You do not have to consider the gaps. So I think the first answer will be correct for this one. So that is all from my side. If you have any questions, you can ask, else we'll drop off. Okay then. Okay, that was what I was showing is yeah, this one you're talking about, right? Can you please confirm? Yeah. So for this, they have asked for the genome assembly size, not the genome size. Right. So the genome assembly size will be the summation of the contig lengths. So you need to work on the contig lengths for they are in geometric progression. So you have to find out the contig length, total summation of the contig lengths, right? You do not have to consider the sorry, gaps and the arithmetic progression. You do not need to consider this portion of the problem because this is given just to confuse you. If you are asked the genome assembly size, it is just the summation of the contig lengths, not you do not have to consider the gap lengths. So if you do that, you'll get this answer. I think this was the correct one. I confirmed with sir also. So this is kind of a trick question that they have put. So instead of genome size, they have asked for genome assembly size. So that will be just the addition of the contig lengths, not the gaps. You do not have to consider the gaps for this. Right? Is that okay? Yeah. Okay. Okay then, guys. Thank you for joining. See you next Saturday, same time. Thank you. Dropping off.